Hi, Julia.
Hello, good morning.
How are you?
Very good.
Good.
You?
Good, thank you. So we have a number of attendees that are joining. Welcome, everyone. We're just giving a few more minutes for more people to join. Still give it 2 minutes or so. Good morning. Welcome to the webinar this morning. We're just going to wait another minute for some more attendees to join. We still have some attendees joining. Okay, seems to have stabilized there a little bit. So I think I'm going to get started just for the sake of time and for everyone who has joined on time. We don't want to waste your precious time. So thank you very much for joining us this morning. My name is Megan Coetzee and I am the product manager for IDT at Whitehead Scientific. We will be recording this session as with the previous two, and at the end of the three-part series, we will be sharing the recordings with you should you wish to revisit them. Also, if you have any questions at the end of the session or during the session, please feel free to type them into the Q&A box. We will answer these at the end of Christos' presentation. Or alternatively, if we don't get a chance to answer them today, we can send you an email as well. So this is the last topic in our three-part series, xGen NGS Amplicon sequencing. And today that is going to be presented by Christos. And Christos, I don't think I'm going to try and say your surname. So Christos is part of the European team of IDT's field applications specialists or scientists, and he supports customers in their technical queries. Christos has a PhD in oncology from the Karolinska Institute in Stockholm, and he has extensive experience with clinical diagnostics based on NGS. So thank you very much, Christos. I will be handing over to you.
Thank you very much. Thank you for the invitation, Megan, and thank everyone that take the time to attend in today's seminar. So I will start already by sharing my screen here. I think you can see already my the website here, and I will share now my presentation. Can everybody see the presentation?
Not yet.
No?
No. Okay, I think
let me try one.
Yes.
I think Lauren is just going to make you the host again.
Yeah.
There you go. You should be good to go now and share.
So let's see here. Share.
There we go.
Now you see the website, yes?
Yes, we can see the website. Thanks, Christos.
Perfect. And now can you see the presentation?
Yes.
Great. So I think we can start. So today we're going to talk about Amplicon sequencing and the Normalase technology. So I will just do a small intro about IDT. IDT, Integrated DNA Technologies, is a company that has a history of more than 30 years in the life science research. We have our headquarters, I will just do my pointer here, in Coralville, in Iowa, in US, and where we have also several R&D and other production facilities. We also have two very big production facilities, one in Leuven, in Belgium, to accommodate our European customers, and also we have one in Singapore for our Asian customers. IDT has a very long history of oligosynthesis and has been involved in the NGS technology for many years now. But what happened last year is that we acquired a company called Swift Biosciences, and we really enriched our NGS portfolio. So currently we're offering solution for library preparation for DNA, and we have kits for normal DNA samples. We have kits for more challenging samples like cell-free DNA and degraded FFPE samples, as well as single strand or low input samples. We have solutions for RNA library preparation, either again for normal, good quality samples or more challenging like low input or degraded samples. And we have also methyl sequencing library preparation kits. And of course, what IDT knows how to do very well is we have a plethora of adapters and indexing primers in order, if you want to increase your multiplexing and really reduce your sequencing costs. Another thing that IDT is very known and is focusing the last year is the targeted enrichment, either by hybridization capture or Amplicon sequencing. I think my colleague Julia in the last session went through thoroughly about the hype capture applications. And today we're going through together the Amplicon sequencing as well as our library normalization kit. I want to also share here that we have a variety of pre-designed panels, and we can help you to have a very customizable solution by having a custom panel, either for hype capture or Amplicon sequencing. Just to start a little bit about the Amplicon sequencing, as we know, both approaches, both hype capture and Amplicon sequencing, come with their benefits. What is the main benefit of the Amplicon sequencing is it's a very fast and easy workflow for you to really simplify the library prep and reduce your costs of sequencing. So how our approach is working is a workflow of around 2 to 3 hours, and we start with a multiplex PCR with one tube PCR, where you are going to amplify your target sequences. And then we're going to continue with a second PCR where you're going to do your barcoding. It's an indexing PCR where you're going to add your UDIs, your unique dual indices, and then your sequencing after bit purification is ready to be normalized and be sequenced. So now we have an optional step of the Normalase in order to normalize your sequencing with a much faster way that I will talk a little bit more thoroughly later on in this presentation. So our solution includes what we call overlapping amplicons in one single tube. And what is interesting with that, here we take two pairs of primers that under normal conditions, you would have only what we call the intended amplicons. From your forward one and the reverse one, you would have this amplicon, and from the forward two and reverse two, you would have this amplicon here. But with our solution of overlapping amplicons in one single tube, you are going to produce two more amplicons. And the most important is this super amplicon here, that it will take advantage of the forward number one and the reverse number two primer. Of course, also we might have these mini amplicons that they are going to be produced by the forward two and the reverse one. But to be honest, those are amplicons that we don't want. Those include very little information, very little data, and because in the next step, they are going also to be amplified and have their barcoding and adapters, they are going to attach on your flow cell. They would take place like a big part of the flow cell, and you don't want that. So we have a proprietary technology to really reduce or almost eliminate all these mini amplicons. You see, under a normal condition, you will have the majority will be mini amplicons, because during a PCR, as you know, the smaller the amplicon, the more privileged is to be, amplified. But with our technology, you almost eliminate the mini amplicons and you end up with the super amplicons and the intended amplicons. And where this technology comes even more beneficial is that a normal downside with amplicon sequencing is that if you are going to have a point mutation underneath an amplicon primer binding site, this primer will not be able to anneal, and then you are going to lose this amplicon here. So this is not good. But with our technology, you're not going to lose any data, any information, because even if you have a mutation here and you lose this amplicon here, you're still going to get your data by the super amplicon that it will be created from the forward one and reverse two primer. So this is a very robust technology, and that's what we took advantage when we created also our COVID xGen sequencing product, because we were able to overcome all the point mutation that they were created with the new variants, the Delta, the Omicron, and always be able to have a complete genome sequencing. I just want to mention here that we have a variety, as we said, of pre-designed amplicon panels, several focused on the oncology and on the inherited disease. We have an EGFR also kit. We have kits for sample tracking. We have very popular kits for metagenomics, where we're sequencing the full 16-length gene, and also for ITS1. We have, as I already mentioned, a SARS-CoV-2 amplicon panel and a brand new xGen monkeypox virus amplicon panel. And of course, as we mentioned before, you can create a custom-made amplicon panel to accommodate your needs. I will just spend 1 minute to talk a little bit about our xGen SARS-CoV-2 amplicon panel. That was a very popular product the last 2 years. We used our overlapping amplicon panel in one tube in order to target the whole 29.9 kb viral genome. And also we had a separate one for the xGen, for the spike protein. And it was a very popular kit because it was very robust with all the new variants, the new mutations, while other approaches like the Arctic or the Midnight panel, they had to make newer versions. Now, if I'm not mistaken, in the Arctic, we're at version 4.1 in order to accommodate this amplicon dropout that it was created by the new mutations from the new variants. We have demonstrated a complete genome from samples with a CT value lower than 33 cycles. And we, at this moment, we can sequence all current variant variants. Another thing that I wanted also to point out is that the Arctic approach and the Midnight approach, the workflow lasts around 7 hours, while with our approach, it is a workflow of around 2 hours plus 1 hour if we want to add the included Normalase step. At this point, I wanted to also explain how you can create your own Amplicon panel. So I will just stop my presentation for 1 second here, and I will go to our website. Can you see my website? Just to be sure that.
Yes.
Yes, yes.
Perfect. Great. Thank you, Megan. So we have here the option to go on product and services. You can go to Amplicon sequencing, and here you're going to come to 2 options, the pre-designed Amplicon or the custom Amplicon. You can go here at the custom Amplicon option, and then it will give you again another 2 options. One to create a new Amplicon panel, and another one to order an already pre-designed panel that you have designed previously. So here you can go to the design requests. You have to log in. Give me a second. And then it will require some information from you. It will require some files to upload in order to do your panel. So you give some general information, and then you will upload here the form with the sequences. You have to choose the species. You have to choose the product. I will talk a little bit more in a bit about the HS product, and then our design team will come back to you with information about your custom Amplicon panel. Here, I want also to say that we have the options of functional testing. That means that we run the prep and we do shallow sequencing, and we check if we have a very even coverage. And if something doesn't look good, we can redesign and tweak a little bit the design to make it very even. So now I will go back to my presentation. Here, that's exactly what I showed you now. And also this order my design option is if you have already a design that you designed previously, you can go there, you can give the design ID, and you can proceed with the sequencing of your panel, with the ordering of your panel. Here, I want to mention also another product that now as a pre-design, it comes only for our EGFR panel, but also you can request that as a custom panel, is the option when you want to reach a variant allele frequency below 1%, then we can take advantage of the UMIs, those unique molecular identifiers, in order to really reach very low in this variant allele frequency percentage. So, you know, when you have specifically samples that they are, for example, like a tumor that is very heterogeneous, so the percentage of the actual cancer cells is very low, so unique molecular identifiers can really help you to reach very low variant allele frequencies. So in this case, we can add 1 more PCR of 3 PCR cycles in order to do the UMI incorporation. And then we continue as previously with 1 multiplex PCR to amplify our target. And then, of course, an indexing PCR in order to add our barcoding. And as I said, this can be also requested as a custom panel. Here are some general information. We provide these panels from an input from 10 to 25 nanogram. We say here as low as 1% of allele frequency for a normal kit and below 1 for our HS, for a high sensitivity kit. It's good to remember that the panel size ranges between 15 and 1500 amplicons. So if you want something much bigger than that, then we have the solution, as Julia said in the last session, of the hybridization capture, where you can go very big in your panel. And these approaches can give an on-target percentage more than 90% with a very good coverage uniformity, and they are compatible with Illumina platforms. So now I will leave a little bit this part, and we're going to talk a little bit more about the Normalase technology. So Normalase technology is an enzymatic normalization technology that really can help you save time and increase your throughput. It can really help you to reduce your sequencing cost, but really balancing your libraries and be able to multiplex much higher in every run. And also it has a very flexible design. So this Normalase is incorporated in our Amplicon panels, but it can also be purchased like a standalone separate kit to be combined with other library prep approaches. So how we actually condition our libraries is that during our indexing PCR, we don't use the normal indexing primers, but we use Normalase compatible indexing primers. So while you do your workflow, your prep, nothing really changes. So you just exchange like the normal indexing primers with Normalase compatible indexing primer. It's very easy, very fast, and for the moment, it's not provided for PCR-free approaches because the conditioning is happening during this indexing PCR step. Of course, there are some minimum requirements, so we prefer if we have at least 12 nanomolar of libraries, but we have a special workflow if you have 6 nanomolar. During the Normalase step, what you do is that you normalize everything in 3 folds lower. So if you start with 12 nanomolar, you are going to normalize all your libraries at 4 nanomolar. If you are going to start with 6 nanomolar, you are going to end up normalize everything at 2 nanomolar. So just to put it a little bit more in perspective, what you're normally going to do after your library prep is ready, you're going to quantify your libraries either with a qubit, quantit, or qPCR. Then you're going to determine the fragment size, fragment analyzer, tape station, bioanalyzer. Then you have to calculate, analyze your data, and then you start the process of diluting and normalizing your libraries. And this can be actually quite time-consuming. And after this step, you are ready to go for sequencing. If you take advantage of the Normalase technology, after your library prep is ready, you can go through 2 enzymatic reactions of around 15 minutes, and then you are ready to go for sequencing. Just to explain a little bit more the workflow, what you're going to do, you're going to prepare a master mix that you're going to distribute in each of your libraries. You're going to have an enzymatic reaction of 15 minutes for on 30 degrees. And then after that, you do a pooling of equal volume. So you take 5 microliters of each of your libraries, and then you pull them all together. And then you have only one tube where you're going to add the Normalase 2 master mix. Then again, you have a short incubation of 15 minutes at 37 degrees. And then you're going to have all your libraries at 4 nanomolar ready to be sequenced. So as we mentioned before, this is a much easier and straightforward option of normalization, skipping qubit, qPCR, bioanalyzer. It normalizes by molarity versus mass, and it's very specialized design for NGS workflows. It's really scalable, so it's a very useful option for high throughput labs, and also it's automation friendly if this is something that it would be interesting. It's so simple that you can add a very easy script at your automation platform. And just to show here some data, here we have some post-amplification qPCR results where we put our threshold, as we mentioned before, at 12 nanomolar. And then we use these libraries to be normalized either by the Normalase technology or by using qPCR and doing our own dilutions and normalizations. Here you have the reads in both cases, and you can see that the coefficient of variation is much lower when you used our Normalase approach in comparison with the mainstream qPCR approach. So this is from me. It was a little bit shorter presentation this time in comparison with your previous sessions, but feel free to ask any questions either on this or the previous information that you got from us in IDT.
Okay. Lauren, can you just
should I maybe stop
okay.
sharing?
Sorry, Christos, I was still on mute there. for a second. There we go. There we go. My camera's on. Okay, so that is the end of the session. Do we have any questions, perhaps? I'm just checking our Q&A. No questions at the moment.
If there are also questions about anything else that they heard during these sessions, because I know that Julia had so many things to present that maybe it was a little bit more.
That's true. If there are any questions from any of the other sessions, you're also welcome to post those here. We have Julia on the call as well. I don't see any questions. Let's give it another minute.
Very often we get the question, what is the best approach for enrichment and why people prefer like height capture or over Amplicon sequencing. So it comes to, first of all, as we mentioned, like the size, for example, of the panel. If it is something smaller, it's easier and faster to do with an Amplicon sequencing, but it's something much bigger, like, we have options like for the whole exome. So then this is something that an Amplicon panel cannot accommodate. So then you have to go for a height capture solution. Then it comes also the part that the Amplicon sequencing is much faster. It takes, as we said, 2 to 3 hours, 2 hours to prepare the libraries, 1 hour to do your normalization, while the height capture, because it has a minimum hybridization step of 4 hours, but very often we recommend to be like overnight, like 16 hours, to be sure that you are capturing as good as possible your target. It takes for sure an extra day. So those are some things that it's good to consider when choosing an approach of enrichment for your applications.
Thanks very much, Christos. Okay, so we don't have any other questions or any questions that have been raised. So thank you very much. A special thank you to Christos and Julia for joining us over the last 3 sessions, for sharing some of the xGen products which IDT has to offer. We really appreciate it. And if you have any questions for the IDT application specialist, please feel free to reach out to anyone at Whitehead who might be your best contact. We also have an IDT in-house application specialist as well, so we will definitely be able to assist you. All right, I hope everyone has a great day further, and thanks again for joining us.
Thank you very much.